Thank you, Christina, for the introduction. I am very happy to be here today, and I wanted to thank the organizing committee for having me here. And today we're going to talk about HIV and how we can intervene in the microbiome to improve the life of people living with HIV. And I'm going to start with this amazing photography where we can see HIV in yellow entering a CD4 cell. And instead of giving numbers about how the HIV is behaving in Europe, which is the context where I am now, I would like to take this opportunity to remind all of you to get tested for HIV in case you have unprotected sex, because the main issue now with the diagnosis of HIV is late diagnosis. Late diagnosis means that you are having HIV for a long time and it hasn't been detected properly. So please remember to get tested for HIV at least once a year if you have risk sexual relations. Now I will talk a bit about the HIV natural history. So once HIV enters the organism, viral load increases rapidly, and the viral load reaches a high level until we detect it and we start antiretroviral therapy in our patients, which would allow us to improve the counts of CD4 cells and other lymphoid cells in the patients. Unfortunately, antiretroviral therapy today works wonderfully, and people living with HIV reaches really old ages. And it is now that we can study these old populations that we can see that they have an increased risk of non-AIDS severe adverse events, such as cardiovascular issues, neoplasia, and higher mortality than the healthy population. This is because HIV infection is an inflammatory pathology, and this inflammation comes from the persistent replication of HIV in blood despite effective antiretroviral therapy, the reactivation of endogenous viruses such as cytomegalovirus or others, and the one that we are more interested in is microbiome translocation that affects the gut-associated lymphoid tissue, which leads to an immune activation which is persistent, and at the end, early senescence and immune exhaustion that would lead to progressive immunodeficiency and a higher risk of these adverse events that I have commented on. And this is because the microbiome influences the immunopathogenesis of HIV and also educates the immune system of our people with HIV. And as I have commented, this has the consequence of increasing the risk of non-AIDS events in our population of interest. And is it possible to influence the microbiota in order to lower the risk of this population of having these events? We don't really know, and many interventions have been proposed, and the one that we used in this project is intervening in it through fecal transplantation, and the hypothesis of this project is that it would reduce systemic inflammation. We used a stool bank in the United States named OpenBiome, and the boss of our group did a hard trip to the United States to basically import high-quality poo from American people, and he brought us to Spain, which was really hard. I mean, red tape was really a big problem here, but after he could do that, we selected a profile of microbiota that would be different from the one that people with HIV usually have. This is basically a high proportion of Faecalibacterium bacteria, a low proportion of Prevotella, and a high proportion of butyrate producing bacteria in the donors. Once we found this nice profile of microbiota, we would give it to the patients. And this project was started by a former colleague of our lab, Alba Talavera, and the design of the project was basically recruiting 30 people with HIV on antiretroviral therapy with some room of improvement in their CD4-CD8 ratio, which is an indicator of immune status in the patients. And we didn't select only one donor of the microbiota, but we selected 3 in order to also analyze the variation due to different stool samples for the transplant. We took samples from the plasma and blood of the patients, and we also took stool samples along different weeks. First, the transplant was done up to 8 weeks, and after these 8 weeks, we also took samples at week 12, 24, and 48. And in this former study, we saw that FMT induced changes in the structure of the gut microbiome. We saw different differences in the alpha diversity and a moderate engraftment of the microbiota, which was most robust in the case of the Lagnospiraceae and Ruminococcaceae families. And also we analyzed different inflammation markers in blood, and we saw that the intestinal damage marker, IFAP, improved in the FMT group. And this was important because this is also an independent predictor of mortality in our population. After these results, we wanted to test if there were more inflammatory protein markers in blood that would be also decreased after FMT, and we used a really cool technique called proximity extension assay, but all in Proteomics in Sweden, which basically detects with 2 antibodies a set of 350 proteins that would then be measured by next generation sequencing as they have covalently bonded a group of oligonucleotides that would come close in proximity and then would be amplified by PCR and further read by NGS. The results of this project were that we saw a complex pattern of protein expression in plasma, and we also saw two main different groups of proteins identified in our samples. By week 24, the patients who received FMT tended to cluster together when compared to the previous weeks and showed lower concentrations of the first group of proteins. And overall, we saw a shift in the inflammation pattern of these patients, which persisted through week 24. We analyzed each of the proteins independently, and we compared the number of significantly different inflammatory proteins, and we saw that by week 24, the FMT group had 12 underexpressed proteins, while the placebo group only had 2. Furthermore, to take advantage of the longitudinal design of the study, we identified 56 differentially expressed proteins using generalized additive mixed models, and most of these proteins, all except 1, were underexpressed in the FMT group. We did also functional analysis of this list of 46 proteins, and we saw that they were mainly involved in cytokine-to-cytokine receptor interactions and also regulation of different immune processes. Now we wanted to know if these inflammatory proteins correlated with the abundance of bacterial species that we quantified by using shotgun sequencing and use linear mixed models, again, to take advantage of the longitudinal design of the project. So we saw 385 bacterial species that correlated with the changes in these 46 inflammatory proteins, and the strongest correlations were observed in species from the Firmicutes families and some others that were previously described in the literature for HIV population. The order Clostridiales was the one that correlated with a higher number of inflammatory proteins, and the changes in proteins such as FLT3LG, IL-12, and others correlated with a really high number of bacterial species. Next, we move to analyzing the correlation of these inflammatory proteins with genes that came from this profiling of the bacterial samples, and we did a similar statistical analysis than the previous one. In this case, we saw strong correlations in the genes that you see in the list, and those were involved in the biosynthesis and transport of metabolites, mainly modulating antigen recognition and contributing to this anti-inflammatory environment that FMT is producing in our patients. Again, we saw the same proteins coming up with correlations with at least 15 bacterial genes. Finally, we wanted to go one layer deeper, and we saw that this decrease in inflammatory proteins correlated with more than 700 bacterial functions that we computed with KEV annotations, and the most relevant ones were related to detoxification and stress response, degradation of fungal and bacterial compounds, pectin degradation, which was nice because this is a short-chain fatty acid production molecule, and also the transport of fructose and bacterial cell wall components. Again, we saw that proteins interleukin-1RN, IL-17A, and IL-6, which are important because they are related with HIV pathogenesis, correlated with the presence of at least 24 functional annotations. Finally, when analyzing in detail these 3 different correlation studies, we propose this method where we would come from a state of HIV before having the fecal microbiota transplant, where we would have a really complex mixture of proteins that would be driving this inflammatory response in the patients, and by doing the FMT, which would be modulated by bacteria, genus, genes, and some specific functions that I have explained before, we would drive the patients from a less inflammatory, from a more inflammatory state to a less inflammatory state because of the FMT. So from this project, we would see that when a patient comes to the clinic, we would profile their microbiota, and if in case they had a pro-inflammatory state microbiota, we would tell our lab technicians to go to our stool bank in the hospital and giving them a transplant with an opposite microbiota profile from the pro-inflammatory one. Although in these projects we are not aiming for that, but we want to understand better which specific molecules, such as proteins or metabolites that are coming from the bacteria, that would really have the desired anti-inflammatory effect in our patients. So as take-home messages, I would like to say that fecal microbiota transplantation from selected donors really resulted in a shift toward a less inflammatory profile. FLLT3LG, IL-17A, IL-6, and IL-10RB may serve as biomarkers for microbiome-related inflammatory responses, and species from Rumnicocacea, Succiniprovignaceae, Proboscidea, and Clostridium genus could be used in targeted interventions. And the most important one is that our study supports in-depth research into the microbiome as a viable target to improve systemic inflammation in people living with HIV or ART, and also other inflammation-related diseases. Finally, I would like to first thank to all the study participants in this study, the different collaborators, and of course, my research group here in Madrid, and to my supervisors, Sergio and Elena, for their support always.